Welcome to our journey into the fascinating world of molecular cloning. Today we'll break down what molecular cloning is, why it's important, and how it has revolutionized biological research. Whether you're a student, a curious mind, or someone interested in biotechnology, this video will guide you through the basics and beyond. Let's start by understanding the core idea. Molecular cloning is a set of techniques used to create identical copies of a particular DNA sequence. This process allows scientists to study genes in detail, produce proteins, and even develop new medicines. So what makes molecular cloning so powerful? Let's find out together. Before diving deeper, let's quickly revisit what DNA is. DNA, or deoxyribonucleic acid, is the hereditary material in almost all living organisms. It carries the instructions needed for growth, development, and functioning. Think of DNA as a biological blueprint, encoding the information that makes each organism unique. Understanding DNA is crucial because molecular cloning revolves around manipulating these genetic instructions. By isolating and copying specific DNA segments, scientists can explore how genes work and how they affect living things. Now let's see how this knowledge sets the stage for cloning. Molecular cloning has a rich history that dates back to the 1970s. Early pioneers developed techniques to cut and paste DNA, opening up new possibilities in genetics. The first successful molecular cloning experiments allowed scientists to insert foreign DNA into bacteria, which then replicated the new genetic material. This breakthrough paved the way for genetic engineering, biotechnology, and modern medicine. Today, molecular cloning is a routine tool in laboratories worldwide, enabling discoveries that were once unimaginable. Let's explore the basic steps involved in this process. Molecular cloning involves several key steps. First, scientists identify and isolate the DNA segment they want to study. Next, they use special enzymes to cut the DNA at precise locations. The chosen DNA fragment is then inserted into a vector which acts as a carrier. This vector is usually a small circular piece of DNA called a plasmid. Once the recombinant DNA is ready, it's introduced into a host cell, often a bacteria. The host cell then multiplies, creating many copies of the inserted DNA. Each step is carefully controlled to ensure accuracy and success. First step in molecular cloning is isolating the DNA fragment of interest. Scientists use various methods to extract DNA from cells, such as chemical lysis or mechanical disruption. Once the cells are broken open, the DNA is purified and separated from other cellular components. This purified DNA can then be analyzed and prepared for the next steps. Accurate isolation is crucial, as any contamination can affect the success of the cloning process. With the DNA fragment in hand, scientists are ready to move forward. Restriction enzymes are essential tools in molecular cloning. These specialized proteins act like molecular scissors, cutting DNA at specific sequences. By choosing the right restriction enzyme, scientists can precisely snip the DNA where they want. This allows them to isolate the desired fragment and prepare it for insertion into a vector. Restriction enzymes are highly specific, ensuring that only the target DNA is cut. This precision is what makes molecular cloning possible and reliable. Vectors are DNA molecules used to transport genetic material into host cells. The most common vectors are plasmids, which are small circular DNA found in bacteria. Plasmids can replicate independently, making them ideal for cloning. Scientists insert the DNA fragment into the plasmid, creating recombinant DNA. Other types of vectors include viruses and artificial chromosomes, each suited for different applications. Choosing the right vector is crucial for the success of the cloning experiment. Once the DNA fragment and vector are prepared, the next step is ligation. This process uses an enzyme called DNA ligase to join the DNA ends together. DNA ligase acts like glue, sealing the fragment into the vector. Successful ligation creates a recombinant DNA molecule, which contains both the original vector and the inserted DNA. This step is critical, as it determines whether the DNA will be properly expressed in the host cell. Transformation is the process of introducing recombinant DNA into a host cell. In most cases, bacteria are used as hosts because they grow quickly and are easy to manipulate. Scientists use methods like heat shock or electroporation to make the bacterial cell membrane more permeable, allowing the plasmid to enter. Once inside, the bacteria begin to replicate, producing many copies of the recombinant DNA. This step is essential for amplifying the DNA and studying its effect. After transformation, not all host cells will contain the recombinant DNA. Scientists use selection and screening techniques to identify successful clones. Often, the plasmid vector contains a marker gene such as antibiotic resistance. By growing the bacteria on selective media, only those with the recombinant plasmid survive. Additional screening methods like blue-white screening help confirm the presence of the inserted DNA. This ensures that only the desired clones are used for further study. Once the desired clones are identified, the next step is amplification. The host cells are cultured in nutrient-rich media, allowing them to multiply rapidly. As the cells divide, they replicate the recombinant DNA, producing large quantities for analysis. This amplification is crucial for downstream applications such as sequencing, protein production, or further genetic manipulation. The ability to generate many copies of a specific DNA sequence is one of the main advantages of molecular cloning. After amplification, it's important to verify that the cloned DNA is correct. Scientists use techniques like restriction analysis, PCR, and DNA sequencing to confirm the identity and integrity of the insert. These methods ensure that the DNA fragment has been properly cloned and that no unwanted mutations have occurred. Accurate verification is essential for reliable research and successful application. Molecular cloning has countless applications in scientific research. It allows scientists to study gene function, investigate genetic diseases, and develop new therapies. By cloning specific genes, researchers can produce proteins for medical use, create genetically modified organisms, and explore the mechanisms of life at the molecular level. The versatility of molecular cloning makes it a cornerstone of modern biology. One major application of molecular cloning is the production of proteins. By inserting a gene into a suitable host, scientists can produce large amounts of a specific protein. This technique is used to manufacture insulin, growth hormones, and other therapeutic proteins. The ability to produce proteins in the lab has transformed medicine, making treatments more accessible and affordable. Molecular cloning is also used to create genetically modified organisms or GMOs. By introducing new genes into plants or animals, scientists can enhance desirable traits such as disease resistance or improved nutrition. GMOs play a significant role in agriculture, helping to increase crop yields and reduce the need for chemical pesticides. This technology continues to evolve, offering new possibilities for food production and environmental sustainability. Gene therapy is an exciting application of molecular cloning. By delivering healthy genes into a patient's cells, scientists can treat or even cure genetic disorders. Molecular cloning enables the production of the necessary DNA constructs for gene therapy. This approach holds promise for conditions like cystic fibrosis, sickle cell anemia, and certain types of cancer. While challenges remain, gene therapy represents a new frontier in medicine. Molecular cloning allows scientists to study gene function in detail. By cloning and expressing specific genes, researchers can observe how they influence cell behavior, development, and disease. This knowledge is essential for understanding the genetic basis of health and illness. Molecular cloning provides the tools needed to dissect complex biological processes and uncover the secrets of life. DNA libraries are collections of cloned DNA fragments that represent the genetic material of an organism. Scientists use molecular cloning to create these libraries, which serve as valuable resources for gene discovery and analysis. By screening DNA libraries, researchers can identify genes associated with specific traits or diseases. This approach has accelerated the pace of genetic research and discovery. Sequencing is the process of determining the exact order of nucleotides in a DNA molecule. After cloning, scientists often sequence the DNA to confirm its identity and study its function. Advances in sequencing technology have made this process faster and more affordable. Sequencing cloned DNA provides insights into gene structure, mutations, and evolutionary relationships. With great power comes great responsibility. Molecular cloning raises important ethical questions, especially when it comes to genetic modification in gene therapy. Scientists and policymakers must carefully consider the potential risks and benefits of these technologies. Open dialogue and responsible research are essential to ensure that molecular cloning is used for the greater good. Working with DNA and living cells requires strict safety protocols. Scientists follow guidelines to prevent contamination, protect themselves, and ensure the integrity of their experiments. Proper lab attire, sterile techniques, and careful handling of materials are all part of safe molecular cloning practices. These measures help maintain a secure and productive research environment. Molecular cloning is a powerful tool, but it's not without challenges. Sometimes DNA fragments don't ligate properly, or host cells fail to take up the recombinant DNA. Contamination, mutations, and low transformation efficiency can also pose problems. Scientists troubleshoot these issues by optimizing protocols and using advanced techniques. Overcoming these challenges is part of the scientific process. Technology in molecular cloning has advanced rapidly. New enzymes, improved vectors, and automated systems have made the process faster and more efficient. Techniques like Gibson assembly and CRISPR-based cloning have expanded the possibilities of genetic engineering. These innovations continue to drive progress in research and biotechnology. Automation has transformed molecular cloning. Robotic systems can now perform repetitive tasks like pipetting, mixing, and colony picking with high precision. This reduces human error and speeds up the workflow. Automated platforms allow scientists to process many samples simultaneously, increasing productivity and enabling large-scale experiments. CRISPR technology has revolutionized molecular cloning. By allowing precise editing of DNA, CRISPR makes it easier to insert, delete, or modify genes. This powerful tool has accelerated genetic research and opened up new possibilities for medicine and agriculture. CRISPR-based cloning is now a standard technique in many labs. Synthetic biology combines molecular cloning with engineering principles to design and build new biological systems. Scientists use cloning techniques to assemble genetic circuits, create synthetic organisms, and develop novel biomolecules. This interdisciplinary field is pushing the boundaries of what's possible in biotechnology and medicine. Molecular cloning has a profound impact on medicine. It enables the development of vaccines, diagnostic tests, and targeted therapies. By understanding and manipulating genes, scientists can create personalized treatments for a variety of diseases. Molecular cloning is at the heart of many medical breakthroughs. Beyond medicine, molecular cloning is used to address environmental challenges. Scientists engineer bacteria to clean up pollutants, produce biofuels, and recycle waste. These applications demonstrate the versatility of molecular cloning and its potential to benefit society in many ways. Molecular cloning plays a role in forensic science. By amplifying and analyzing DNA from crime scenes, scientists can identify suspects and solve cases. Cloning techniques help ensure that even tiny amounts of DNA can be studied, making forensic investigations more accurate and reliable. Molecular cloning is an essential part of biology education. Students learn cloning techniques in the classroom and laboratory, gaining hands-on experience with modern biotechnology. This training prepares the next generation of scientists to tackle new challenges and drive innovation. Public understanding of molecular cloning varies widely. Some people are excited about its potential, while others have concerns about safety and ethics. Education and transparent communication are key to building trust and ensuring that society benefits from advances in cloning technology. Governments and regulatory agencies play a vital role in overseeing molecular cloning research. Regulations ensure that experiments are conducted safely and ethically. Compliance with these rules protects both researchers and the public, fostering responsible innovation. The future of molecular cloning is bright. Emerging technologies such as gene synthesis and advanced automation promise to make cloning even more powerful and accessible. As our understanding of genetics grows, new applications will continue to emerge, shaping the future of science and medicine. Personalized medicine tailors treatments to an individual's genetic makeup. Molecular cloning enables the development of targeted therapies based on a patient's unique DNA. This approach holds great promise for improving outcomes and reducing side effects in medical care. While DNA cloning is the most common, scientists also clone RNA and proteins. These techniques expand the range of research possibilities, allowing for the study of gene expression, regulation, and protein function. Cloning beyond DNA opens new doors in molecular biology. Molecular cloning is transforming agriculture. By cloning genes that confer resistance to pests or harsh environments, scientists are developing crops that are more resilient and productive. This technology helps ensure food security and supports sustainable farming practices. The production of biopharmaceuticals relies heavily on molecular cloning. By cloning genes that encode therapeutic proteins, scientists can produce medicines that treat a wide range of diseases. This approach has led to the development of life-saving drugs and vaccines. Molecular cloning is used in industrial biotechnology to produce enzymes, biofuels, and other valuable products. By engineering microorganisms, scientists can create efficient production systems for a variety of industries. This technology supports innovation and economic growth. Studying cloned genes helps scientists understand evolution. By comparing DNA sequences from different organisms, researchers can trace evolutionary relationships and uncover the history of life on Earth. Molecular cloning provides the tools needed for these important discoveries. Molecular cloning is playing a role in conservation biology. By cloning genes from endangered species, scientists hope to preserve genetic diversity and support species recovery efforts. This approach offers new hope for protecting biodiversity in a changing world. Molecular cloning enables the development of synthetic vaccines. By cloning and expressing specific antigens, scientists can create vaccines that are safe, effective, and easy to produce. This technology has been vital in responding to emerging infectious diseases. Diagnostic tests often rely on molecular cloning. By producing specific DNA or protein probes, scientists can detect diseases quickly and accurately. Cloning technology has improved the sensitivity and reliability of many diagnostic tools, benefiting healthcare worldwide. Synthetic gene construction is a cutting-edge application of molecular cloning. Scientists design and assemble genes from scratch, enabling the creation of entirely new biological functions. This approach is driving innovation in biotechnology and synthetic biology. Despite its many advantages, molecular cloning has limitations. Technical challenges, ethical concerns, and regulatory hurdles can slow progress. Scientists continue to refine techniques and address these issues, ensuring that molecular cloning remains a valuable tool for research and innovation. Molecular cloning has transformed science, medicine, and industry. By enabling the precise manipulation of DNA, it has opened up new frontiers in research and innovation. As technology advances, the potential of molecular cloning will only continue to grow. Thank you for joining us on this journey through the world of molecular cloning. Stay curious and keep exploring the wonders of science.